All right, I guess we'll get started. It's a Friday after all, right? There's kind of two semi-separate topics in today's lecture. The Cambrian explosion stuff, which is this follow-on of the history of life that we were talking about. So it's kind of the particular course of events as appears to have happened as known to scientists. And then, and most of that stuff doesn't have lots of kind of conceptual information. There's some stuff to learn and think about, but the second set of topics about macroevolution and different kinds of processes in macroevolution. God, that's where there's some kind of conceptual nuggets that I want you to try to understand as processes and concepts that are more general to the entire process of evolution than just kind of what happened on Earth so far as we know. So, as a recap, there were cells roughly 3.5 billion years ago. They resembled cyanobacteria. They probably used DNA as the information-carrying molecule. And then life marched on for another over 3 billion years, basically as single-celled organisms. So there's this, if you think about the span of time of Earth history from when the Earth was formed, roughly 4.5 billion years ago, to cellular life, that takes about 1 billion years, and then life stays as simple unicellular kinds of organisms for a little bit over 3 billion years, which is really kind of remarkable span of time. But what happened in that 3 billion years, by life staying unicellular, you shouldn't conceive of that as there not being a lot of evolutionary change going on. There's lots of evolutionary change going on in biochemical pathways and things like that you don't necessarily see at the kind of macroscopic level of organisms. So lots of change is happening, but it's not particularly visible. Multicellular life, larger organisms, that's what we're more able to see in the fossil record. So that's what's called the Phanerozoic, which is visible life, meaning macroscopic. So these are multicellular organisms. They appear as fossils, roughly 565 million years ago. And over the next 40 million years, you get great diversification of the types of these fossils that are found, and to the extent where essentially all of the animal phyla that we recognize now have evolutionary precursors recognizable in these fossils that are, between 565 and 525 million years old. So that's this kind of supposedly sudden appearance of a huge amount of biological variation, right? You go from 3 billion years of unicellular organisms to a 40 or so million year period where you get multicellular life apparent in the fossil record, and you get the origin of the extant phyla of animals. So that sudden appearance is called the Cambrian explosion. So this period, is called the Cambrian, is when you have these first visible forms, and the explosion part is because of this apparently sudden appearance in the fossil record. So as you look through what paleontologists have been able to find out about the fossil record, you kind of get to start to appreciate that level of diversification in that period of time. A bit about the fossil record and the kinds of things that paleontologists study. You're all used to sort of the Natural History Museum kind of giant dinosaur skeleton. That kind of fossil is the permineralized bone, where there's bone that the bone structure has been replaced by mineral material. That's kind of the classic fossil. But there's lots of other kinds of fossil things which are just include things like impressions. If you have really soft, squishy mud and something sticks into it, like a footprint, for example, that would be an impression-type fossil. When we talk about human evolution, there's really famous human footprints in volcanic ash. That is, there's no fossilized material of the humans, but you have this sort of negative space impression that shows you that something was there, and it shows you the gross morphology of it. Permineralized fossils are these kinds of things, dinosaurs, and these are fossil trees. But you also get casts, which is kind of like the inverse of an impression. The cast is the interior impression. So if your skull was cracked open, you could get a cast of the inside of your skull. That's how one way that people study brain case evolution in humans is by the impressions of the inside of the material. Again, the fossil doesn't have any bone or structure that was formerly bone, but it sort of reveals what those structures were from an interior kind of view. And then for more recent things, and especially good for insects, amber, which some of you might like as kind of jewelry. Amber is essentially a fossilized tree sap. Kind of looks like this sort of stuff. It's this yellowish golden thing, and when trees are oozing sap, most of you probably know that lots of insects get stuck in it. It's pretty sticky stuff. So there are a lot of really well-preserved fossils of things, but especially insects and other arthropods that are within amber deposits from different places around the world. Isn't that supposedly in Jurassic Park that I forget exactly the story. What, they found a mosquito in there? Dinosaur DNA in the blood, inside the mosquito, inside the amber, inside or something like that, right? That's the story of Jurassic Park. All true, I'm sure. Okay. So the thing about the fossil record that you kind of have to live with is that it's extremely imperfect, and it's not imperfect in a random kind of way. It's imperfect in ways that are sort of systematically biased in terms of what kinds of organisms were present. So you would like a fossil record that would preserve a lot of the history of life and preserve it in a way that represents that total biodiversity over time, and it doesn't. Most things that live don't get fossilized, and there's systematic bias in the kinds of things that do get fossilized. So you're much more likely to have representatives of certain kinds of things than other kinds of things. So it's not only incomplete, it's biased. So the likelihood of fossils being created depends a lot on the durability of the kind of material we're talking about. So that's why you don't typically get fossilized tissues. You're much more likely to get fossilized bone. Even more common than fossilized bone is fossilized teeth. Even more common than that is fossilized shells, right? Hard, durable materials that are likely to be buried when the organism dies. If they're likely to get buried, they're likely to be into an anoxic environment where they're not going to decay. So lots of, you're much more likely to get fossilization with marine organisms, or lowland deltas are river systems that are dumping sediments on top of the kinds of fossils. So they're the kinds of conditions that favor the burial of whatever died. So there's really good fossil records for some things like clams and other marine organisms with hard shells, and just terrible fossil records for other kinds of organisms that are soft, squishy type things. Things that live in hot, wet, humid, tropical type environments are much less likely to fossilize than things that live in river basins or marine environments, or hot, dry environments where they're less likely to decay too. So it's very imperfect and biased, but you still get a lot of information about what kind of stuff was there, even though it's incomplete. So if you remember back to our talk about the age of the Earth and the different kinds of assemblages of organisms, this idea of geological time, before there was radioisotope dating, there were still these different geological time periods that were identified by fossil assemblages. So in this strata, you always find these kinds of organisms, and below it, you always find these other kinds of organisms, and you find other things up here in this other kind of stratum. That's how it was originally divided up by these fossil assemblages, and only later were they dated by radioisotopes. There's this kind of hierarchically nested way of dividing up time, where the big things are eons that are divided into eras, that are divided into periods, that are divided into epochs, kind of like, orders and families and genus and species, sort of. This way of dividing up life, this Phanerozoic period that we're talking about, in this scheme, the Phanerozoic is an eon. So in common language usage, we say, oh, it was an eon, just meaning it was really a heck of a long time. But in paleontology, it has a specific kind of meaning. The Phanerozoic is divided into these three eras, the Paleozoic, Mesozoic, and Cenozoic. Everyone knows what the root zoic means, right? There used to be zoology departments that studied animals. Zoic is, this is referring to animal life. Paleo, anybody? All right, let's start easy. Meso. Meso's in the middle. Paleo's old. Ceno is recent. Not quite new. But so the old life, the middle life, and the more recent life. Sometimes when you go to natural history museums back in the day, these used to be divided into what were called, the age of fishes, the age of reptiles, and the age of mammals. That's total nonsense. It is and it has always been the age of insects. You know that, right? Yeah, okay. Whatever. So here's Paleozoic. Whenever I put these things up in class, I always get these questions like, what are we supposed to remember from this? Blah, 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 blah. Is that going to be on the test? How do you spell it? All that kind of stupid stuff. There's a lot of interesting stuff here that you should try to kind of absorb. But I have a memory like a sieve, so I tend to focus on concepts and ideas rather than very specific kinds of information. I think it's more fulfilling to do that. If I forget this stuff, I can look it up. It's not especially intellectually gratifying. But you should have this sense of, Paleozoic, about when did it start, right? About when did it end, So the Paleozoic starts with the start of the Cambrian. Here's the Cambrian. So you have this Cambrian explosion at the start of the Paleozoic, roughly 550-ish million years ago, and it ends with the Permian at the huge extinction events that characterized this changeover of fauna. So the end Permian mass extinction events that we mentioned the other day, and we'll talk about again today, that's what characterizes the sort of end of the Paleozoic. And you should have some appreciation of the kinds of things that occurred. So, for example, we already mentioned the Cambrian explosion has the ancestors of almost all animal phyla appearing. So you have shelled organisms, arthropods, chordates, bryozoans. You have early jawless fishes, land plants, insects, ferns, vascular plants, winged insects, tetrapods. There's a lot of stuff that are the kinds of organisms that you would recognize in your world now. They're obviously very different in their specific details, but plants, vascular plants, seed plants, insects, these kinds of things are appearing in this general time period. The other thing you should have some appreciation of is the sort of biogeography of this, how major land masses have moved around, and the Earth physical structure in the Paleozoic, at any rate, was really, really different from what you would recognize today. There's main ones is that you have these supercontinents of Laurasia and Gondwana that sort of contained the kinds of continents that we recognize now, but they were arranged and packaged together very differently, which had a very profound effect on the biogeography of organisms that we see now. And we don't talk that much about biogeography in here, but you should at least appreciate that these things are very different and changing over time. This is where all your fossil fuels come from, your oil and coal and this stuff, is these things that are buried. Okay. Mesozoic, the second age of insects. This is dinosaurs. So you have another mass extinction event that marks the kind of beginning of this kind of transition. You have dinosaurs. You have, this is a kind of more interesting point, the first mammals and the first dinosaurs appear very similar sorts of times. But throughout the Mesozoic, it's the dinosaurs that diversify and occupy the large land vertebrate niches. So the big herbivores are plant-eating dinosaurs. The big carnivores are meat-eating dinosaurs. The mammals during this time are these little kind of shrew, mouse-ish-like things that are running around and not occupying the kinds of ecological niches of big predators and big herbivores and stuff like that, even though they're roughly coincident in origin. 65 million years ago, dinosaurs, right? That's the end of the dinosaurs. That's the end of the Mesozoic. Huh?
In the Paleozoic, it says about seed plants and vascular plants, and then in the Mesozoic, it says that gymnosperms become dominant, and then angiosperms. Does gymnosperms not appear until the Mesozoic?
So what would be non-gymnosperm seed plants? I think they are. I think they are. The plants that I know of are, the seed plants that I know of are gymnosperms or angiosperms. So I think it's early sort of, or maybe the ancestors of gymnosperms.
Not ferns.
Not ferns, right. So ferns reproduce with spores, mosses and bryophytes reproduce with other things, but gymnosperms are pines and cycads and stuff like that don't have these fleshy seed coverings. So I think it's probably this gymnosperms dominate, then first flowering. This is angiosperms. So yeah, I'm not sure exactly how they're dividing up these pre-gymnosperms, but they're the ones that I know of, seed plants. All right, so Cenozoic, dinosaurs are wiped out, and you get this radiation of mammalian orders. So the mammals were there throughout most of the Mesozoic, but were not dominant, did not diversify, were not occupying these terrestrial, lots of these large terrestrial organism kinds of niches. It's only after the dinosaurs are wiped out that the mammals radiate and become, the Cenozoic age of mammals kind of thing. Another kind of interesting thing here, we're just talking about flowering plants. The angiosperms are the flowering plants, and the pollinating insects are radiating. Radiating basically means diversifying. Lots and lots of new species are being created, And the fact that these are coincident things that are both occurring at the same time, you have this radiation of flowering plants and the radiation of their pollinators, suggests that these things are co-evolving and radiating sort of in response to each other. There's pollinator specificity of insects and the kinds of flowers that they pollinate. So there's some kind of suggestion of actual process behind this, not just stuff to try to memorize. Let's see, what else happens that's noteworthy? You get apes. You get the earliest hominids. We'll talk about that stuff later. You have this Antarctic ice cap and worldwide glaciation. You know, it's cold. You get the start of the Pleistocene, the North and South American land bridge that we talked about the other day in terms of these Alpheus snapping shrimp that are in the Pacific and Caribbean side. That's sort of when this division happened, roughly 3 million years ago. So lots of cool stuff in the history of life. You're certainly not going to absorb all of those details, but big picture stuff, do your best. Okay, so rewinding back to the Cambrian, this 40 million year period when these major animal taxa appear, one thing that as biologists you should appreciate, well, what makes phyla different? Why do we say these different phyla are appearing? How are we recognizing them as different? And what's distinguishing this diversification of organisms really early in multicellular life? And the main things that distinguish these different phyla are their embryonic development and the adult body plan, how the body is packaged together. And when you guys watch this YouTube video from PBS Nova about these transformations of organisms, there's segments in there about body plan, evolution, and the genetics of it, and you should pay attention to body segmentation and how that kind of thing evolved and works at a mechanistic genetic basis. But at the non-mechanistic genetic basis, the major kinds of things that divide animals into phyla are whether they're diploblasts or triploblasts, the type of coelom they have, which is the gut cavity, whether they're embryonic development, that gastrulation is if they're protostomes or deuterostomes. So we're going to kind of have a quick tour through what those things are, see if you can figure out which you are as you go along. So diploblasts have two types of embryonic tissue, an endoderm and an ectoderm. What does derm mean? Skin. What does endo mean? Ecto? Little geniuses. Okay, these organisms are radially symmetrical. What does that mean? Their axis of symmetry is round. You can cut them in, oh, I should switch to blue here. You won't see it on a red thing. You can cut them in lots of different planes, and they would be equal size. They have some kind of roundish type of body plan. Triploblasts have 3 embryonic layers. They add a mesoderm. Again, meso means middle, right? And they're characterized, not all of them, but they're characterized by bilateral symmetry, which means that they have a left side and a right side. So lateral means side. So which are you? You're triploblasts. I could be mean and say some of you are rounder than others, but that's, no, I won't do that. I wouldn't be that mean. Okay, so within the triploblasts, there are different types of gut development. Acoelomates, this prefix A means they don't have it. It means it's not there. So things like flatworms don't have this kind of gut differentiation. Coelomates have this true gut. that's derived from the mesoderm. The pseudocoelomates have a body cavity in them, but it's not homologous to the body cavity of the coelomates. It's a different embryonic development. It's not from the mesoderm, and it's secondarily derived. You are a very nice. Okay. Here's two triploblast coelomates, just like you. One of those things is much more closely related to you than the other. Which are you more closely related to? A lot of people might guess the crab because it has bilateral symmetry, like you do. But crabs are actually protostomes that when this ball of cells gastrulates, so gastrulation is when it folds in to form the gut. You fold in the mouth end and you fold in the butt end, and that gets the butt that's, the tube kind of gut. If you do the mouth first, that's a protostome, and that's what crustaceans and lots of other stuff is, like these crabs. If the mouth comes second, you're a deuterostome, and that's what echinoderms are, as well as you. So you're more like this, even though these have radial symmetry. Okay, so that's kind of where you fit phylum-wise. What's kind of neat about the Cambrian explosion is there's two really well-preserved sets of fossils that are more early Cambrian and more late Cambrian. So they're about 40 million years apart. One's found in Australia called the Ediacaran, and it's about 565 million years ago, and it has these soft bodied impression fossils. There aren't bones or shells or teeth or anything like that. Sometimes it's just the fossils are kind of tracks. They're just in the mud. What's the symmetry here? This is radial symmetry. There's squishy, soft kinds of things: sponges, jellyfish, comb jellies, and then some 40-ish million years later, there are deposits that were first found in British Columbia called the Burgess Shale, and then the same fossil assemblage of organisms was later found in Xinjiang, China, and they're impression fossils, but you can see these have bilateral symmetry, and you have these whole different kinds of sets of organismal body plans. So the presumption is that sometime within that 40 million year period is when you get the development of these large variation in body plans. But again, the molecular machinery behind that variation in body plans was evolving for many, many, many millions of years before this kind of macroscopic diversification took place. So when you use molecular data to try to figure out this Cambrian explosion stuff, so you can date the fossils and the rocks using radioisotope kind of dating, and you get these 550-ish, 565-ish million years old. If you just use DNA sequences among living organisms, so you take living organisms, you analyze their DNA sequences, and you infer using a molecular clock how long ago would that divergence event have been, to how much time would it have taken to get that level of divergence in the molecular sequences. The dates that people get, they're obviously variable and subject to error and that kind of stuff, but they're consistently a lot older than what you get from the fossil dates. So, for example, you get dates approximating 1 billion years of molecular divergence, maybe twice as much. So there's, in other words, the discrepancy between the molecular data and the fossil radioisotope datings is more than just the measurement error of those kinds of estimates. The molecular divergence appears to have preceded, by a large degree, the morphological stuff that you see in the fossil record. And there's two kinds of possible explanations for that. One is there are older fossil assemblages or were older fossil assemblages that we haven't found that we might find or we might never found. And the other nearly equally plausible explanation is that the molecular machinery is diverging long before the macroscopic body plans are diverging. And when you look at the video about transitions and body plan evolution, and what I hope you see out of that is that you can get really major changes in the body plan features of organisms which would appear at this kind of level, with relatively minor changes in the timing of expression of genes related to development, but that the origin of that genetic variation and developmental stuff could easily precede and is likely to precede their expression morphologically. So that would go some ways towards reconciling those differences. So stay tuned. Research continues. So kind of maybe more satisfying question to ask was, well, why did this happen? What are the kinds of selective forces that promote this kind of seemingly abrupt diversification? And one thing to realize is that there's big changes in the Earth kind of environment and atmosphere and the kinds of ecological resources that organisms can diversify to exploit. So we've talked several times about ecological niches being the way that organisms make their living, and we've talked about specialization and diversification, and specialists being better able to exploit discrete different kinds of resources. So if you start early in the Cambrian with the Ediacaran fauna, they're basically filter feeders. They're not moving around. Sessile means they just stay put. They're just sitting there, right? There's not locomotion. And then over this 40-ish million year period, you develop grazers and predators and burrowers, things that are moving, that there's locomotion, where these things are actively foraging and moving about. And one of the things that may have favored that is rising oxygen levels in seawater. So you have all this cyanobacteria churning out, photosynthesizing and churning out oxygen. The levels of oxygen that fuel metabolism that would allow for locomotion, those were increasing in the Earth's atmosphere. And then when you make this big switch from being a sit-there filter feeder to a moving around kind of thing, if you're moving around, you can move around and graze, as in eat algae and eat cyanobacteria and that kind of stuff, but you can also move around and eat other things that are moving around, right? You can be a predator of those kinds of things. If predators on things favor defenses and escape mechanisms, all of these kinds of things, having locomotion, having movement, favors all kinds of traits about organisms that weren't favored before. The most obvious is that locomotion favors bilateral symmetry instead of radial symmetry. If you're moving through a fluid substrate, you don't want to be round. That's an extremely inefficient way to push through some kind of medium. If you're pushing through some kind of medium, if you're moving, one part of you is going to get there first, right? All of a sudden, you have selection for a head end and a back end. You would put, well, the part of you that gets there first is probably where you want to concentrate your sensory organs. You want to know what's there when you get there before the rest of you gets there. So having sensory organs on the front end, having a mouth organ on the front end is favored. These are major body plan kind of things that, in a sense, they're so obvious that you don't think about them. But if you're moving from these radially symmetric filter feeders that don't move and don't go anywhere and do anything, there are actually really big changes in the body plans of organisms. So a head-end sensory system on the front end, having predators favors defenses and hard body parts like shells and stuff like that that fossilize more easily. So you get really big turnover in the kinds of stuff that's there because of this shift from sessile planktonic filter feeders to things that move around, some of which are sort of like herbivores and some of which are kind of like carnivores as predators. That favors all kinds of different things. And then this is kind of a theme we've talked about, the really big part of the selective environment then becomes other organisms. It becomes that biotic environment selects for lots of further changes rather than just the ability to deal with salt water and cold and that part of the abiotic environment. So when you have coevolution between different parts of the biotic environment, you get even more specialization and diversification. So that's cool stuff. So that's kind of leading into this sort of part 2 of the lecture, which is much less about the specific history of life and hopefully has more kinds of conceptual principles that might apply repeatedly to different kinds of episodes of life evolution.